In this topic, we will be discussing about the patterns of distribution of the organisms in on the surface of the earth. So first topic is the finding a home. That means how an organism finds its habitat where it is living. So the aim of biogeography is to understand underlying patterns of distribution of organism on our planet. But these organisms are living on a particular habitat, and they are found on that particular habitat due to some reason. Such studies must be based on the appreciation of the nature of species with which one is dealing, right? So when we talk about the organism, we talk about a particular species, maybe. We talk about a particular genus, taxa, any taxonomic distribution, we study about that. But when we talk about an organism, we normally think of a species, a particular group of organism who is able to interbreed and produce the fertile offspring. And where that species is present, or what is the history of that species, we need to discuss. So some species, and then there are the after the species, then there are the subspecies. So some species, for example, exist in a number of different forms that are sufficiently stable to termed as subspecies. So further level of species is the subspecies, two organisms which are having such similarities that they could be counted as one species, but they are living in different areas and having different acquired characteristics as well. So these species are called subspecies and have and these often have different distribution patterns. So normally we divide the subspecies on the basis of their distribution patterns. There are the polytypic species and monotypic species. A species that exist as a series of subspecific forms is termed as a polytypic species. So poly means many, type from the type comes the typic, so that means that it contains many types, and as opposed to the less variable species that exist just in one form, and they are called the monotypic species. So how do we know that one species is different from another, or one subspecies is different from another despite their different distribution patterns? We do the genetic analysis. Biologists apply the genetic analysis, and these more widely explain that how the species distribution takes place. The complex relationship within the species are becoming increasingly evident, and such complexity is reflected in the distribution patterns. And then there is the example of Larus argentatus. It is an example of polytypic species. And the name of this organism is the herring gull. Gull is the waterfowl, and it is present in different northern areas of the world. It was regarded as the polytypic with about a dozen subspecies spread around entire northern hemisphere, and it is the distribution which is termed as circumboreal. So northern hemisphere is above the equator, all the areas towards the northern side. So here you can see a picture of approximate breeding distribution of various taxa within the herring gull complex. One is the European herring gull, two is the American herring gull, three is the yellow-legged gull, four is the Caspian, Hugelian, Tamir, Vega, steppe, Mongolian, and Armenian gull. So there are the different distribution patterns, but these patterns of distributions are not very perfect. These are just distribution factors. But if we see in detail their genetic analysis, many of these will be very much related to the different. For example, the European may be very much related to the yellow-legged gull as well as there might be the 6 and 7, Tamir gull and Vega gull may be very much genetically related. So these are the studies that we can do at a genetic level. And what is the gradual, when a species is going from one distribution pattern to the next or evolving in a different areas, it has some gradual change in it. So where there is a gradual change in the genetics and form along a gradient, taxonomists refer to as a cline. and the variation is said to be clinal. For example, we have seen the gulls. All of those gulls have a clinal changes across the globe. So they are differently going from one place to another, having different distribution patterns. Not only that, but also they have different genetics as well. And then there is another example of evolution of bonobo, a consequence of separate genetic development resulting from the presence of major barrier to the interbreeding, which is River Congo. All chimpanzees subspecies lie to the north or east of the river. So the bonobo and the chimpanzee, they had the common ancestor, but the River Congo divided them into two different groups. One are the chimpanzees, another one is the bonobos. So here you can see the distribution pattern of chimpanzees, subspecies, and bonobo in the Central and West Africa. 1 to 4, that is the north of the Congo River, and that are the chimpanzee, which is pan-triglodytes. And then there is the bonobo or pygmy chimpanzee, which is pan-peniscus. So more specifically, you can see the names of different chimpanzees, subspecies. The bonobo is found on the south of the River Congo, is hence separated from the other chimpanzees by a formidable barrier to the movement. So why the bonobo diverged and had different distribution as well as different species, a completely different species than the chimpanzee, because there was a geographical barrier, which is the Congo River in the Africa. And then there are the microhabitats as well. So such as the forest canopies or forest floors. In New Zealand, forest brown Kiwi Apteryx australis is the forest living on the forest floors, while the another kiwi, which is the fan-tail kiwi, Ripidura fuliginosa, nests in the canopy. So they are having the same habitat, but within the habitat they are founding the different microhabitats. So you see that how the different organisms have evolved to get the different habitats, and why they are found in different habitats today. We study that in the patterns of distribution.